Hello everyone. Today's biochemistry class is on extracellular matrix. In this class, we will know what is this extracellular matrix and its biomedical importance, what are the components of extracellular matrix and what are the functions. To know the different types of extracellular matrix which are involved in the body, which are there in the body, and what are the different disorders associated with extracellular matrix. Cells, we know they are the basic units of the life, and these cells, which are located in the different tissues, are surrounded by a complex extracellular matrix, which is generally called as connective tissue. So this extracellular matrix is nothing but a network of interstitial proteins which surrounds the cells which are located in the tissues. So this is the connective tissue which is surrounding the cells, and it is nothing but network of interstitial proteins. What is the significance of this extracellular matrix, or why we need to know about different types of extracellular matrix is that these extracellular matrices have got different roles, are involved in both normal physiological and pathological processes in the body. That is, since during development the embryonic cells, they have to travel distances through these extracellular matrices to find their ultimate destinations. Even in the inflammatory processes, there are different changes, biochemical changes, which will be observed in the extracellular matrix. In the cancerous tissues, the cancerous tissues, to metastatize or to spread, they have to detach from their original tissue of origin or organ, and then they have to travel through these extracellular matrix and enter the different blood vessels or the lymphatics. There are different genetical diseases or inborn errors of metabolism associated with different extracellular matrix or connective tissues like collagen, elastin and other things. Several autoimmune diseases like rheumatoid arthritis, osteoarthritis, where there is evidence of involvement of this extracellular matrix in such diseases also. So extracellular matrix or connective tissue has got important roles in both physiological and pathological processes. So coming to components of extracellular matrix, it is basically made up of three different types of proteins. The first one is called as structural protein, and it includes collagen, elastin and fibrillin. Here you can see the collagen. The main function of collagen is to provide the strength. The second type of protein which is involved in the extracellular matrix formation is adhesive glycoproteins, and it includes fibrillin, fibronectin and laminin. And the main function of these adhesive glycoproteins is that they connect extracellular matrix elements to one another and also to the different cells. And the third component of extracellular matrix is water hydrated gels known as proteoglycans. Proteoglycans nothing but protein molecules associated with carbohydrates. So it basically consists of long chains of repeating disaccharides attached to proteins, and the main function of this is to permit comprehensive resistance and lubrication. And the another component of extracellular matrix is water. So it is basically nothing but different proteins. Extracellular matrix is nothing but consisting of different types of proteins with different functions or specific functions for each type of proteins. So coming to the functions of extracellular matrices, the main function of extracellular matrix is to provide the support for cell migration and also for the maintenance of cell polarity. It is involved in the control of cell proliferation. It is required for scaffolding for the tissue renewal after the tissue injury. It helps in organized regeneration of the tissues. It is required for the establishment of tissue microenvironments and also required for the maintenance of normal tissue architecture. These are the functions of extracellular matrix. Moving on to the most common type of collagen which we will see, which is present in the body, that is collagen. It is the major component of the most connective tissues present in the body, and it constitutes about 25% of the proteins of the mammals. It provides extracellular framework, stability of the tissues and organs, and it is present throughout the body, and the major fibrous elements of tissues like bone, teeth, tendons, cartilage and blood vessels also. There are basically 19 types of collagen which will be seen in the body. Moving on to the structure of collagen. Collagen is made up of 3 polypeptide chains which is again braided into a triple helical structure, and this polypeptide chain has got about 1000 amino acid residues, and based on the composition of these amino acid, 27 types of different types of collagen are described, and the most common amino acid present in the collagen is glycine. That is about 33% of amino acid present in these polypeptide chain is glycine. That is, every third residue present in this polypeptide chain will be of glycine, and the next commonest amino acids will be proline and hydroxyproline, followed by lysine and hydroxylysine, and rest of the chain contains different amino acids. But the most common amino acid, that is every third amino acid in the chain, is glycine, and the next common amino acid will be proline and hydroxyproline, lysine and hydroxylysine. So this is the sequence. This is the triple helical structure of collagen. Out of 27 types, foremost are the commonestly we will be studying. Type 1 is most abundant and it is present in almost all the regions of the body. Type 2 is seen in cartilage and vitreous humor. Type 3 is seen in skin, lung and vascular tissues. Type 4 is seen in the basement membrane. And anchoring fibrils will contain type 7 of collagen. Beaded filaments will contain type 6 of collagen. And the disorders which are commonly seen will be associated with these type 7 of collagen. Coming to the synthesis of collagen, the steps involved in this collagen will be synthesis, post-translation modification, followed by glycosylation, extracellular maturation and the cross-linking. And the collagen molecule will be synthesized in the form of pre-procollagen, procollagen, tropocollagen and finally the fibrillar collagen. Synthesis, coming to synthesis proper, collagen is synthesized by the intracellular fibroblasts from the ribosomes, and it is synthesized in the form of pre-procollagen. it is taking place on ribosomes by the intracellular fibroblast in the form of a pre-pro collagen. And this pre-pro collagen from the ribosomes, it moves into the vascular space of the endoplasmic reticulum. And in the endoplasmic reticulum, this polypeptide sequence, whatever is there, that will be enzymatically removed, and then post-translational modification will take place. In the post-translation modification, mainly the hydroxylation of proline and lysine residues takes place again intracellularly, and this process requires iron, ferrous iron, molecular oxygen, and most importantly vitamin C. And the enzymes involved in the hydroxylation of proline and lysine residues will be prolyl hydroxylase and lysyl hydroxylase, and procollagen is formed from the pre-pro collagen molecule. And then the glycosylation takes place. Once the hydroxylation of this proline and lysine takes place by the hydroxylase enzymes with vitamin C, ferrous irons, and molecular oxygen, these will undergo glycosylation process, that is attachment of glucose and galactose molecules to these hydroxylated polypeptides with the help of the enzyme galactosyl transferase and glucosyl transferase. After this is completed, the procollagen molecule is secreted outside, that is extracellularly, and in the extracellular compartment, with the help of specific peptidases, the N-terminal and C-terminal procollagens are cut off. About 150 amino acids in the N-terminal area and about 300 amino acids in the C-terminal area will be cut off. And then these tropocollagen molecules will be reassembled as collagen. So any deficiency in these specific peptidases for the cleaving of N and C terminal amino acids will lead to a genetic condition called as dermatopraxis. It means the patient will have soft, duffy skin which is very, very fragile, and at the same time it is saggy and redundant, especially on the face, and the patients may also have hernia and also possibly mild to severe form of joint hypermobility. Once this is done, cross-linkings will be formed between lysine and hydroxylysine residues with the help of lysyl oxidase enzyme, and this requires copper. So any deficiency of copper during this cross-linking of lysine and hydroxylysine residues will lead to a disease called Menkes disease. Deficiency of copper required by the lysyl oxidase for the cross-linkage formation between lysine and hydroxylysine residues will lead to Menkes disease. So this cross-linking will convert amino acids into aldehydes or aldol crosslinks. So just short revision of synthesis of collagen. Collagen is synthesized in the form of pre-pro collagen on the ribosomes, and this will be transferred to endoplasmic reticulum where hydroxylation of proline and lysine takes place, which requires ferrous iron and vitamin C. Once hydroxylated, this hydroxylysine and hydroxyproline will undergo glycosylation by addition of glucose and galactose, and the triple helix procollagen is formed. this procollagen will be transferred outside, and in the extracellular space, cleavage of N and C terminal amino acids will take place with the help of specific peptidases, and tropocollagen is formed. And this tropocollagen later on will form cross links between lysine and hydroxylysine residues and converts amino acids into aldehydes, and this forms the stable collagen fiber. This is about the synthesis of collagen. So degradation, it takes place by the collagenase enzyme, and this collagenase enzyme is synthesized by the Clostridium histolyticum bacteria. So degradation of collagen will be seen more in presence of osteoporosis, tumor metastasis during postpartum period, Paget's disease, rickets, osteoarthritis, rheumatoid arthritis, and also in the scurvy. So functions is mainly to provide the mechanical support and provide alignment to the tissues. Disorders associated with collagen includes osteogenesis imperfecta, Ehlers-Danlos syndrome, Alport syndrome, Epidermolysis bullosa, vitamin C deficiency, Lathrism, and Menkes disease. Collagen synthesis, osteogenesis imperfecta or brittle bone disease. About 30 genes are involved in the encoding of the collagen, and that's, as we learned, the pathway of synthesis is also complex. So there are different genetical disorders which can be associated with the mutation in the genes, and also in the mutation in the genes or absence mutation in the genes or mutation of genes, including the enzymes which are involved in the collagen synthesis. So the first disease which we'll be seeing is osteogenesis imperfecta, also called as brittle bone disease. This disease is characterized by abnormal fragility of the bones, and the sclera of these patients is usually thin and translucent. Over 90% of the patients who have osteogenesis imperfecta will born with multiple fractures and usually not survive. The basic pathology or biochemistry behind this disease is that there is mutation of the type 1 collagen, where because of this the mutation, there will be replacement of glycine by the cysteine in the type 1 collagen. So this leads to disruption of the triple helical structure of the collagen, and fibrillar arrays will not be formed. So all this will lead to brittle bones or fragility of the bones, which causes multiple fractures, skeletal deformities, bowing of the back, and also barrel-shaped rib. So because of so much fragility of the bones, the infants who born with this disease will have multiple fractures and usually do not survive after the birth. Even if they survive, there is no cure for this. Only symptomatic treatment is possible, that is, the fractured bones can be repaired, and again, because of this fragility, the bones will be breaking again and again. The second disease will be Ehlers-Danlos syndrome, and this disease is a group of inherited conditions where principal or the most significant feature or hallmark of this disease is hyperextensibility of the skin and the joints. The defect here is the type 3 collagen formation because of defective lysyl oxidase and lysyl hydroxylase enzymes. So clinical features will include weakening of the type 3 collagen. They will have loose skin, hypermobile and lax joints. The hallmark of this disease will be hyperextensibility of the skin and the joints. The third disease associated with collagen synthesis is Alport Syndrome. Here it mainly affects the kidneys. The defect here is the mutation of the collagen 4A3, collagen 4A4 and collagen A5 genes. The mutation is happening in these three genes, which is linked to X chromosome. Because of this, the type 4 collagen will be defective. So this mutation, since type 4 collagen is mainly present in the glomeruli of the kidneys, so this results in the defective glomeruli, which in turn prevents the kidneys from proper functioning, that is filtering of the blood is not taking place. So this leads to progressive loss of the kidney function. Since basement membrane, the glomeruli mainly needs collagen 4 for the proper functioning. So because of mutation, the collagen 4 is not formed properly. So the basement membrane of the kidney is affected, which leads to progressive loss of the kidney function. Clinical features includes hematuria. Hematuria is the most common symptom seen in Alport syndrome, and patients will eventually develop end-stage renal disease or chronic kidney disease. They may also have lenticonus, that is protrusion of the lens capsule and the underlying cortex, and they may also have bilateral sensory neural hearing loss. This includes the Alport syndrome. First feature will be hematuria, followed by CKD, lenticonus and bilateral sensory neural hearing loss. Wherever type 4 collagen is present, it is affected. Next common disease which will be seen will be Epidermolysis bullosa. It is a non-inflammatory blistering disease. Mutation is taking place in the type 7 collagen, which forms the anchoring fibrils. These anchoring fibrils are very much important for the adhesion of basement membrane to the upper dermis. So, mutation of type 7 collagen will alter the anchoring fibrils. So these blisters will occur just below the basement membrane and will result in significant scarring and milia formation. So here we can see the skin blisters and breaks in the skin formation. Next common disease which is associated with collagen is scurvy. It is not because of deficiency of vitamin C, which is required during the post-translational modification of the collagen synthesis. So scurvy, the patients will present with bleeding gums, and the gums will be swollen, spongy. The pulp will be separated from the dentine and the teeth will be lost finally. Wound healing may be delayed or lost. The capillaries also will become fragile and bleed easily. Spontaneous bleeding is common. Petechiae, ecchymosis and hematomas are also seen. In case of severe deficiency of vitamin C, they may have epistaxis, hematuria and melena. And internal bleeding is also seen in very severe deficiency of vitamin C. Moving on to the next connective tissue, that is elastin. Elastin, it is a connective tissue protein which is responsible for the property of extensibility and elastic recoil in the tissues. Even though it is not widespread as much as collagen, but elastin is mainly present in large amounts, particularly in those tissues which requires this elastic physical property, especially the ligaments, walls of the blood vessels and the tissues. The gene which is responsible for elastin is chromosome 7. Here the major amino acid which constitute elastin is proline, and the polypeptide chains which are present in the elastin will be cross-linked together to form rubber-like elastic fibers, and these fibers will stretch and can resume back to their original length. And this elastin is usually synthesized in a precursor form called tropoelastin, and this is followed by hydroxylation of prolines of the tropoelastin, and again it is formed hydroxyproline. Moving on to the differences between collagen and elastin. Collagen has got different genetic types, about 30 genetic types, and elastin has only one genetic type. Collagen has triple helix, elastin does not have no triple helix. It has random coil formations. It has glycine repeating structure. In elastin, there is no glycine proline hydroxylysine repeating structure. They have to undergo glycosylation, that is carbohydrate containing. The hydroxylysine, after the hydroxylation, glycosylation takes place, but here in elastin no carbohydrates. Intramolecular aldol crosslinks are formed, but in elastin intramolecular desmosine crosslinks are formed. And in collagen, presence of extension peptides takes place during the biosynthesis, but in elastin no extension of the peptides which are present during the biosynthesis. Disorders related to elastin includes Williams-Burnet syndrome because of the deletion of gene which is present on chromosome 7. Here the patient will present with different severe developmental abnormalities in connective tissues almost all over the body. The second one is pseudoxanthoma elasticum. This is again inherited defect in the formation of elastin, and the patient will have loose skin, hypermobile and lax joints. And also a number of skin diseases, for example scleroderma, are also associated with accumulation of elastin. Fragmentation and a decrease of elastin is also found in certain conditions such as pulmonary emphysema and also in the aging of the skin. Next one is the fibrillin. It is a large glycoprotein which is synthesized by extracellular fibroblasts. It is found in the zonular fibers of the lens, periosteum and in the arterial wall. And this also has 4 different types, type 1, 2, 3 and 4. Type 1 is major component of the microfibril. Type 2 is associated with early osteogenesis. Type 3, fibrillin is mainly present in the brain, and type 4 is also associated with early osteogenesis. The disease associated with defective fibrillin is Pierson Syndrome. Mutation in the fibrillin gene 1 will lead to a syndrome called as Pierson Syndrome, and it is autosomal dominant in nature. The clinical features includes ocular changes, skeletal deformities and cardiovascular abnormalities. Ocular changes will include ectopia lentis, that is dislocation of the lens. Skeletal deformities will include long digits, hyperextension of the joints, and in the cardiovascular manifestations, aortic aneurysm, mitral valve prolapse are commonly seen. Next connective tissue extracellular matrix is fibronectin. It is a cell surface glycoprotein and it enhances cell adhesion to extracellular matrix components. And this plays important role in cell adhesion, migration, blood clotting and wound healing. And this fibronectin has got binding sites for other components like collagen, integrin, heparin, fibrin, DNA and to the cell surface. Next is laminin. It is a basement membrane glycoprotein which has 3 polypeptide chains. It has adhesive properties. It enables epithelial cells to fix to the underlying tissues, and it plays an important role in the neuronal outgrowth and nerve regeneration. And increased expression of laminin is seen in the senile plaques and amyloid proteins. And the disorder related to laminin includes progeria, that is early onset of aging, and this may be because of defective laminin. And next disease is Pierson Syndrome, where there is mutation in the gene of laminin B2, and this will be presenting with congenital nephrotic syndrome with ocular and neurological defects. Next one is proteoglycans. Proteoglycans are nothing but combination of proteins with glycosaminoglycans. And the proteins which are associated with these glycosaminoglycans are called as core proteins. And glycosaminoglycans are nothing but unbranched polysaccharide chains which are composed of repeating disaccharide units. And these proteoglycans are of 7 different types, which includes hyaluronan, dermatan sulfate, chondroitin sulfate, heparin sulfate, keratin sulfate 1 and 2, and heparin. The functions of these glycoproteins includes helps in the cell migration and adhesion. It helps anchoring cells to migrating to matrix. It helps in anchoring of cells to matrix fibers and helps in organizing water molecules and resistant to compression, that is in lubrication. And these glycosaminoglycans will be degraded by specific lysosomal enzymes, which includes endoglycosidases and exoglycosidases. So any defects in these enzymes will lead to accumulation of these glycosaminoglycans in the lysosomes, and the condition is called as mucopolysaccharidosis. And there are different types of MPS or mucopolysaccharidosis, includes type 1, 2, 3, 4, 6, 7 and 9. Type 1, 2 are most commonly seen. Type 1 is called as Hurler's syndrome, 2 is Hunter syndrome, 3 is Sanfilippo syndrome, 4th one is Morquio syndrome, 6th one is Lamy syndrome, 7th one is Sly syndrome, 9th one is Natowicz syndrome. And the different glycosaminoglycans are affected, that is accumulation of these particular glycosaminoglycans will take place in different types of mucopolysaccharidosis. So other collagen vascular diseases which are commonly seen is systemic lupus erythematosus, rheumatoid arthritis, Sjogren's syndrome, mixed connective tissue disorders. And the common signs and symptoms associated with these disorders will include fatigue and body ache, muscle weakness, fever, joint pain and skin rash. Localized forms of collagen deposition defects will include, which are commonly seen, hypertrophic scars. Here there is accumulation of excessive amounts of collagen. Keloid formation, when the tissue scar which grows beyond the boundaries of the original wound and it will not regress, and this will lead to formation of keloid. And the third one is Beal syndrome. Beal's syndrome. Here there is contraction of the hip, knees and ankle joints takes place. Moving on to the summary to finish this chapter. Extracellular matrix is nothing but a network of interstitial tissues, and this includes 3 different types of proteins: structural proteins, collagen, elastin, water hydrated glycosaminoglycans, and adhesive glycoproteins. And the basic function of this extracellular matrix is to provide mechanical support, control cell proliferation, scaffolding of tissues for the renewal, and cellular interaction. And many genetic diseases and autoimmune diseases are related to extracellular components. Since the collagen has got 30 different genetic encoding, and also the synthesis of collagen is more complex and different enzymes are involved. Many genetical disorders or inborn errors of metabolism are related to collagen synthesis, and we have seen them. Thank you.